Unlock the future of pathology with Pathology News, your ultimate source for cutting-edge insights, expert analysis, and the latest breakthroughs in digital and computational pathology. Don't just stay informed, stay ahead.
Ladies and gentlemen, it's 10 a.m. in New York and 3 p.m. here in London. Good morning, good afternoon to you wherever you are, and welcome to our webinar today, which is entitled Seamless AI Integration of AIRA Matrix's Tools on HistoWiz's PathologyMap platform. My name is Jonathan Tunstall. I'm from Pathology News, and together with the Pathology News team, I'm pleased to be your host for this event today. Now, as is usual for these sessions, your microphones are muted to avoid background noise, and we will hold a question and answer session at the end of the discussion. Your questions may be posted in the drop-down panel, which you'll see just to the right of your screens, and you can do that at any point during the session. Now, we will attempt to deal with as many questions as possible following the presentation today. Now, I'd like to introduce our panelists, our two panelists. We have Akash Parvatikar, who's a computational pathologist and AI scientist, specializing in histopathology image analysis and spatial tumor heterogeneity using machine learning and graph-based deep learning. He holds a Ph.D. in computational biology from the Carnegie Mellon University of Pittsburgh, and is a product manager at PathologyMap, where he's doing innovation in digital pathology. Our second panelist is Nitin Singhal. He's head of products at AIRA Matrix and is focused on AI-driven medical imaging for predictive diagnostics and personalized medicine. Nitin has expertise in AI, computer vision, and medical image analysis, and he's authored over 50 publications, holds 11 patents, and develops impactful healthcare solutions. So at this point, I'll stop talking and hand you over to our two speakers. And I believe, Akash, that you're taking over.
Okay, great. Well, thank you. And I'm excited to be part of this webinar. We have a lot of interesting discussions that will be part of today's presentation. And I will just get started with the webinar. So it's nice to meet all of you here. And I'm Akash. I'm working as an AI scientist and product manager of Pathology Map platform at HistoWiz. And HistoWiz is a full-service histopathology lab service provider for research with an in-house image management system called PathologyMap platform. We also have a network of over 135 board-certified pathologists who support our telepathology offerings. So what's unique to PathologyMap platform is that we cater to over 4,000 active researchers from over 500 organizations across pharma, biotech, and academic institutions that use HistoWiz not just to produce highest quality digital slides with fastest turnaround time, but they also leverage us for our platform that has helped them accelerate their research. We also produce and house one of the largest and most diverse preclinical data on Pathology Map platform, and this gives us a unique opportunity to host multiple AI models that is compatible across this diverse dataset. We have enabled this by designing Pathology Map as an AI orchestration platform to make it easy for researchers to host their slides and run cutting-edge AI tools. So to support this vision, we are excited to partner with AIRA Matrix, who have a range of validated AI models in the preclinical sector. And with that, I would like to pass it on to Nitin to introduce himself, who is the head of products at AIRA Matrix. Off to you, Nitin.
Hello, everyone. Thank you, Akash. Very nice meeting you, everyone. Before jumping into the slides, I would like to introduce AIRA Matrix. AIRA Matrix is a technology company based out of Mumbai, India. We work primarily in the life sciences space, providing AI-based solutions for diagnostic, prognostic, and prediction. In terms of the verticals, we work in two major verticals supporting the pharmaceutical industry with the assessment of animal toxicity and animal efficacy studies. And on the clinical side, we work on healthcare diagnostics, focusing on prostate cancer, lung cancer, and GI cancers. We are a 120 plus teams with expertise in, of course, AI, looking at product development, domain expertise, and sales and marketing. In terms of our collaborations and clients, we work with the top 5 pharmaceutical companies, CROs, and specialty hospitals like the NHS, and as well as the academic university hospitals in the US and in UK.
Well, thank you, Nitin. And so in today's session, we will discuss what are the high-level benefits of AI orchestration, followed by some of the key features of Pathology Map platform. And next, Nitin will take over and talk more about his company and how the tools are benefiting the research community. After that, I will show you how we at HistoWiz have integrated AIRA Matrix AI tools onto our platform. And finally, this will be followed by a live demo and Q&A. So before we jump into the details of how we have integrated the AI tools on our platform, let's step back for a second and understand what is AI orchestration and why is it important to digital pathology. So imagine a unified space where the pathologists and researchers can seamlessly trial, purchase, and integrate AI algorithms into their workflows without being constrained by IMS compatibility. So, with an ever growing number of AI solutions, Pathology Map acts as a centralized hub that allows direct comparisons, independent user reviews, and a streamlined procurement process that reduces administrative burden. So this not only accelerates AI adoption, but also empowers pathologists to make data-driven decisions with confidence. And on the other hand, for AI developers like AIRA Matrix, it's a gateway to market visibility and it offers a launchpad for further innovation. And yes, challenges in commercial scalability do exist, but with the right approach, AI orchestration is the future of digital pathology, and Pathology Map is leading that charge. So, Pathology Map itself has three main components. We have ordering on the left hand side here, we have slide management, which is the middle, and on the right hand side, we have analytics. So, let's start with ordering. It's pretty quick and straightforward. So our customers place an order either for histology or slide scanning service, or clients upload their digital files directly to the platform. So the ease of use of all of this on the platform ensures that our customers focus more on their research and less on administrative tasks. Next up comes the core of our platform, which is slide management that enables our customers to effortlessly view their slides, organize them, analyze the data, and share all of it with their team members. But that's not all. If our clients need expert eyes on their slides, we provide them with top tier expertise with our dedicated image analysis team and telepathology, which has over 135 board certified pathologists across multiple specialties. And adding to the analysis workflow, is our recently launched AI apps library that allows users to get easy access to cutting edge AI tools from several vendors. So there are several AI solutions out there, but many of the researchers that we have observed with our customer base, they have issues with integrating those multiple AI solutions for their use case. We want to enable our customers to analyze their slides using state of the art AI without installing additional software. And by putting all of these tools directly onto the platform, it makes it incredibly convenient for our customers. So they don't need to source all of these tools. They don't have to integrate it to a specific platform. And they have all the benefits of scalability and security of our cloud provider. And for analysis of the slides on PathologyMap, we have a bunch of AI apps that is deployed onto the platform. We have internally developed automated quality control and tissue segmentation. And further, we have recently integrated AIRA Matrix AI app, and you will be able to access their tools as buttons on the platform. And after this webinar, if you have any questions about how to access these buttons on PathologyMap or how you can upload your digital files, I'm happy to take any questions. Next, I would like to pass it along to Nitin, who will tell you more about AIRA Matrix and the suite of incredible AI tools that they have built over the past few years. Off to you, Nitin.
Thank you, Akash. Great. Today, I'll focus on three key advancements which AIRA Matrix has done. Uh, we are in the business of doing tox assessment, developing AI solutions for tox assessment as well as efficacy studies for more than a decade now. What has enabled that experience is into three key advancements, starting with an AI enabled primary and peer review. Pathologists play a crucial role in ensuring diagnostic accuracy, but traditional workflows can be very time intensive and prone to human variability. Our AI driven primary and peer review systems acts as a co-pilot, enhancing diagnostic precision by flagging potential anomalies, standardizing assessments, and providing second opinions. Secondly, optimized turnaround time for faster study reporting. Speed is critical in pathology. Delays in study reporting can slow down research, drug development, and eventually the patient care. Our AI powered system streamlines workflows by automating quality control, pre screening these slides, and prioritizing critical cases, leading to significant reduction in the turnaround time. Third, innovative anomaly detection framework. Identifying anomalies in pathology images is essential for accurate diagnosis and drug safety assessments. Our AI driven anomaly detection frameworks help recognize subtle deviations in tissue morphology, uncovering insights that might otherwise be missed. By integrating machine learning with pathology expertise, we provide an enhanced data driven reporting. Traditionally, toxicology study assessments have been highly manual, requiring extensive time and effort from pathologists to analyze slides, document findings, and conduct peer reviews. This process is not only time consuming but also prone to variability and human subjectivity. With digital pathology, we introduce AI assisted automation, significantly reducing the turnaround time while improving consistency and objectivity in assessments. AI models are designed so that they can rapidly screen and analyze large volume of whole slide images, flagging potential abnormalities for further review by expert pathologists, and enabling the pathologist to focus on complex cases. As we transition into digital workflows, ensuring the quality of histopathology images becomes paramount. Artifacts such as out of focus region, missing tissue, stain inconsistency, air bubbles can impact the accuracy of diagnosis and reporting. A robust quality control mechanism is essential to detect and quantify these issues, ensuring that only high quality images are used for analysis and reporting. At AIRA Matrix, our suite of AI powered tools is designed to enhance both primary and peer reviews in digital pathology. AI assisted primary reviews help streamline the initial analysis by detecting and classifying key histopathological features, providing an additional layer of confidence for the primary pathologist. On the other side, AI assisted peer review ensures a standardized and an objective second opinion process, reducing both the intra and the inter observer variability. One of the core challenges in digital pathology adoption has been the interoperability, ensuring that AI driven tools work seamlessly with existing image management system and the infrastructure which is there in the pharma company as well as the hospital systems. Our modular design is built with this in mind, allowing for smooth integration with third-party image management solutions like the HistoWiz's PathologyMap. This ensures that institutions can leverage AI-powered pathology tools without disrupting their existing workflows. In preclinical research and drug safety evaluation, accurately detecting and quantifying toxicological abnormalities is critical. Manual assessment methods can be time consuming, subjective, and prone to human subjectivity. That's where AIRA talks come in. AIRA talks has been specifically designed to assist pathologists by automating the detection and quantification of abnormalities in histopathology images. This allows for a very objective, data driven reporting that enhances the reliability of toxicology studies. By leveraging AI, we can quantify structural changes in tissues, ensuring that findings are consistent, reproducible, and backed by comprehensive evidence. One of the key advantages of IRATOX is its ability to standardize toxicological assessments across studies and institutions. This ensures that the results are not only faster, but also more transparent, helping to support regulatory submissions and decision making with robust digital evidence. IRATOX is built to address key challenges in hepatotoxicity, cardiotoxicity, reproductive toxicity, looking at both male and female reproductive toxicity, immunotoxicity, as well as nephrotoxicity. At the heart of IRATOX are 150 plus supervised learning models. These models have been meticulously trained to detect, classify, and quantify toxicological abnormalities, ensuring an evidence based approach to pathology review. What makes IRATOX truly groundbreaking is its preclinical foundation model, a robust AI framework that has been trained on a massive dataset of more than 130,000 images. These images have been collected from multiple laboratories across various species and tissue types. This comprehensive training allows IRATOX to generalize effectively across diverse toxicological studies, making it versatile and highly reliable tool for preclinical research. Pre-trained AI models have been developed to detect, classify, and quantify histopathological features across multiple organs. Let's take liver as an example. The AI models in our platform can automatically identify and quantify key histopathological features, including hepatic vacuoles, fibrosis, necrosis, single cell necrosis, looking at inflammations, looking at bile duct hyperplasia, and few other abnormalities. Similar algorithms have been available for kidney, heart, lung, spleen, some of the reproductive organ, brain, looking at immune organs like thymus, lymph node, and spleen. As we push the boundaries of drug development, ever increasing need for faster, more reliable, and unbiased methods to identify potential toxicological risk early in the pipeline. Traditional supervised methods, while effective, rely heavily on manual annotations, which can be time consuming, prone to variability by the pathologist who are annotating this data, and often struggle in case of rare abnormalities, those subtle and rare abnormalities where not much data is available. This is where unsupervised anomaly detection plays a game changing role. Instead of relying on predefined labels and human annotation, these AI driven models learn pattern directly from the normal control data, identifying deviations that may signal potential toxicological risk. The power of this approach lies in its ability to handle high throughput screening efficiently. With thousands of whole slide images processed daily, the AI rapidly scans and prioritizes slides for expert review, reducing the workload for pathologists while ensuring no critical findings are overlooked. This not only accelerates decision making, but also enhances the consistency and objectivity, eliminating any potential human bias in early stage toxicological assessment. Furthermore, by integrating these unsupervised anomaly detection models into preclinical workflow, we can detect novel toxicological patterns that might have otherwise gone unnoticed, offering early warnings on compound toxicity before clinical trials, ultimately leading to safer drug development. We present a fully automated AI powered pipeline, beginning with high throughput screening and leading to detailed identification and quantification of individual abnormalities. The process starts with unsupervised anomaly detection, which enables the high throughput screening. And this process eliminates the need for exhaustive manual review of every samples, allowing the pathologist to focus on the most relevant samples. Once the AI detects potential slides with abnormalities, the pipeline advances to the identification and the quantification stage. These supervised deep learning models precisely classify and measure specific histopathological features. To conclude, I want to highlight the key takeaways about how AIRA Matrix is revolutionizing the non-clinical toxicological workflow with IRATox. IRATox is built to optimize workflow efficiency by seamlessly integrating into existing toxicological pipelines, enabling the pathologist to handle high throughput studies with greater ease. It reduces manual workload, automates complex pathological workflows, and enhances decision making through AI powered insights. The platform streamlines pathology workflows by automating triage and accelerating reporting. Instead of manually reviewing every slide, IRATox prioritizes the most critical cases, allowing pathologists to focus their expertise where it matters the most. Its AI driven analysis ensures precise, consistent, and objective assessment across large data sets. Thank you. Over to Akash.
Thank you, Nitin. So with HistoWiz and PathologyMap, our main goal with launching AI apps on the platform is to make it easy for users to run and visualize AI result. And Nitin went over some of the AI tools across multiple different organs and species that AIRA Matrix has developed, and also get as much information about these tools for our users to make sure it's relevant to their research. So for that, we have the compute page on our platform, and this is where you can access all the information about your slides, what AI tools are available, and view results for the tools that you have already processed. You can think of it as a dashboard to essentially monitor what specific tools you ran on your images and further take action on your slides. And again, we will go over this in detail when I give a demo of the platform. So AI orchestration platforms like PathologyMap has several advantages, which we saw early on. And one such advantage is that it simplifies invoicing and contracting by offering a single point of transaction for multiple AI tools. Now, this eliminates the need for separate agreements with each vendor. And this not only reduces administrative overhead for pathology labs, but it also streamlines procurement and ensures faster access to cutting edge AI tools without the complexities of managing multiple supplier contracts. So, this is how the AI apps library looks like on the platform. And it lists all of the AI models that is available on PathologyMap. And here you can run any of it by simply clicking on the run model button. And again, we will discuss this in more detail when I give the demo. But essentially, you can scroll through any particular app and visualize the result directly on the slide viewer. And also, you can request for quantifications and generating reports, which we will talk about in a couple of minutes. Here on the right hand side, you can see information about a specific app, which includes access to validation reports, such as like on what data the model was trained on, what is the accuracy, and things like that. It also gives you some information about what organ and species is the AI model compatible on. And of course, also, it also includes the image format compatibility. Say you might be working with CZI files or DICOM. So you want to make sure that the AI is compatible on those image formats. So we want to give as much information uh, before you use a specific app for your research and analysis. So next, for every single analysis that you do using the AI apps on the platform, you will have access to overlay visualizations, which you are seeing here. And for demonstration, say I clicked on the liver analysis app from AIRA Matrix. it populates with these overlays here, which you can see in blue, which represents bile duct. And not just that, it also gives you access to quantifications. And here you are seeing that 0.5% of the entire tissue area has bile duct, spanning across over 1700 regions, across the entire whole slide image. Again, information like this is useful to accelerate pathology discoveries, because as you know, identification might be easier compared to quantifying it, and accurate quantification, and that's where AI helps to do that. And not just that, you also have the ability on the platform to download the raw annotations. Again, why? Because there might be few pharma companies or biotechs who have their custom analysis workflows once they get the AI result. So we also make it possible to download the raw annotations, and this could be embedded into your analysis workflows for downstream processing. So with that, we will switch to a live demo of PathologyMap, where I will go over some of the critical features of the platform and talk in more detail about how we have integrated AIRA Matrix app and how you could run it, and how do you access all the information about the app itself. All right, so this is the pathology map platform from HistoWiz. At a very high level, what you're seeing here is a data management dashboard that essentially gives you a visibility to what orders you have placed in the past. And the data management button gives you access to information as to how much data you have on pathology map across what slide types. It could be bright field H&E, immunohistochemistry, or immunofluorescence images. And we have the compute button, which I showed during my presentation. And this gives you access to details such as what AI app you ran on a particular slide, along with the order ID and the slide name. And say, for example, you have run liver analysis from AIRA Matrix. So these two buttons here, again, I will come back to that. So when you click on liver analysis button over here, it takes you to the app page, which gives you all the information about the validation reports or demo video and things like that. And but if you click on the view result, it directly takes you to the slide viewer, where you can easily access the AI result from that particular app for that slide. Again, I will show both the app page and the slide viewer in a couple of minutes. But before that, we have made it very easy to upload files directly to the platform. So you can create an account on HistoWiz app. And once you have done it, you can just click on the upload button, and you can either select an existing project or create a new project. And for example, say I selected this project, I click next, and again, you can either create a new order or upload slides to the end-to-end existing order. And once you click on next, it generates a link for you to upload your files directly. Again, one takeaway from this is Pathology Map not only supports your traditional SVS and TIFF files, but we also support NDPI, CZI, DICOM, OMITIF, and all in all, we support close to 28 file formats. Now, once you click on proceed to upload, it takes you to a page wherein you can directly upload your files either one by one or an entire folder. And you will be notified once the slides are uploaded, and then you can come back to the platform and look for the images. Now, once you have images on the platform, there are a couple things that you can do to use the platform itself. And one such feature is our search tool on Pathology Map. Now, this tool has been incredibly popular with our pharma, biotech, and academic clients. And what makes this unique is that you can filter across hundreds of thousands of slides in milliseconds. And that's not even an exaggeration. So every image that you're seeing here, the file size ranges between anywhere between several hundred megabytes to several gigabytes. So the team has spent a lot of efforts to make sure the experience is quite seamless to search through multiple slides. Now, way the search works right now is based on the metadata. And for example, you want to look at liver, rat across H&E stain type. So I have selected these three metadata fields. You can easily search across all your images that matches this particular metadata. Now, what you're seeing here is the data that the clients have contributed to HistoWiz. So we have the use rights for it, but this experience will be exclusive for you. So what I mean by that is, say you have uploaded 10,000 images across multiple organs and species. So the search experience is exclusive to the slides that you have uploaded. And again, say you click on a particular liver rat sample, you can click on the slide viewer here, and you can quickly zoom in and zoom in all the way to a 40x resolution very rapidly, and look for any anomalies, or it could be pathology consultation that you might be interested in. So the takeaway here is that the experience on PathologyMap is quite fast when you're trying to zoom in, zoom out, and pan across these giant file-sized images. Again, all of this, as I mentioned, is based on the metadata that you would have entered while uploading your images or while placing an order with HistoWiz. But we also have the ability on the platform to easily update the metadata. So what you're seeing here is a giant spreadsheet which has all the information about your project, orders, slides, and the associated metadata. Now, there are some pre-populated fields that you might have entered, but say as your research evolves, you want to create new fields. So you can easily do that by creating a new category. Say, for example, I'm interested in bile duct, and percentage bile duct itself would be a metadata field. Now, the way in which you can populate these fields could be done in two ways. One is you can import a spreadsheet with that information, and we do a simple sample name to sample name matching and populate it for you. Or if you have used AI analysis on the platform directly, so we automatically populate the metadata based on the analysis. So again, for example, if you're interested in some of the liver features from AIRA Matrix, such as percentage bile duct, percentage vacuolation, etc. So you can run the AI app from AIRA Matrix and then add it as a metadata, and then that becomes a search field. So you can easily search through your liver tissues, which sort of exceeds a certain threshold for bile duct or vacuolation. And up next, the platform itself supports customizable hierarchy. So we have several pharma customers and biotechs who have an exclusive way in which they want to organize their data across projects, orders, and images. So the platform allows you to do that. And not just that, it also allows a high level customization through slide groups. So what this essentially means is it's like a Spotify playlist. So where, if you are creating a playlist, you can essentially take in songs from within an album or across albums and create a playlist. So similarly over here, you can pull in images within your order or multiple orders and multiple projects, uh, put it together, create a slide groups which is easily shareable with a simple link. And we have customers who have directly used that link in their publications. Again, an easy way for a reviewer to look for the images on which their manuscript is based on and things like that. So now, again, this is also one of the very popular tools that the customers have been using, especially in the research space where there is a lot of collaboration happening uh, between the teams, within the teams. And again, the platform doesn't have any limitations on number of users who can onboard. There is no limit over there. So uh, you can easily, add in multiple users, create the slide groups, and then share it with your research collaborators. So say, for example, I have created a group here called as IRA liver analysis demo. And once you click here, the share group is on. So what this means is now I can share these group of slides. I can just click on the link, and you can share this link to anybody who also doesn't have an account with HistoWiz. So They can log in as a guest user and they can have access to the slide viewer which you are seeing. Now, coming back to the slide viewer. So here what you're seeing is the Pathology Map platform's slide viewer, which has several different features. We have slide measurement features, which allows you to measure between regions within the tissue. And we have annotation features that lets you annotate, with a free polygon or a square and add comments to that. Again, all of these annotations and comments is also easily shareable along with the images. Now, for every image, we have some critical information that you can see. One is the metadata, which I spoke about when I was talking about, how you can search the metadata and modify it. So you can view the metadata for every single image. And what's interesting is that you can also look for, which tissue block did that slide come from. Again, this is not relevant if you have uploaded slides directly to the platform. And again, I won't go in detail about this feature, but HistoWiz has a block management solution that gives you visibility as to what tissue block it came from if you digitize slides with HistoWiz. And up next, so this is the slide list that lets you access multiple slides simultaneously. And coming back to the AI apps library. So this is the suite of AI tools that we have right now on HistoWiz, and we will be continuing to add more and more tools. Uh, for example, AIRA Matrix. For now, we have integrated liver analysis tools, but we are interested to integrate more apps because I think they champion in preclinical research space, and again, it aligns with the wide customer base that we have at HistoWiz. Now, we also have few internally developed AI tools. We have tissue segmentation tool, and you can control, the way in which you want to set the transparency or to visualize the AI result. We have an internally developed QC tool. And again, this tool helps us streamline our histology workflow. So it was critical for us to build this tool to accelerate our end-to-end histology service and, again, sort of enhance producing high quality data set for our customers. So as a part of that, we have out of focus to detect tissue blur, cover slip, tissue dirt, tissue cracks, etc. And once you click on any of that feature, it populates with bounding boxes where green indicates there is no issue and red indicates there's a significant issue. And again, tissue blur is something, which is not clearly visible at lower resolution. So an AI app for that actually, makes it easy to look for anomalies across the image. For example, here it flags that there is one region which has blur, and as you zoom in at a very high resolution, indeed there is a small blur within the tissue. Again, it depends on researchers as to how critical is a QC issue for them. But not just for the researchers, even for AI companies, QC has been incredibly useful, because they want to run their AI on good quality regions. And if for a certain reason it gives X percentage of accuracy or AUC, they want to know that it has come from the good quality regions within the image and not the bad ones. And coming back, as I mentioned previously, we have also integrated third-party AI companies like AIRA Matrix. And the way in which the AIRA Matrix AI tool works is that basically you can click on Run Model over here. This is changed to Order Details for the purpose of demonstration because we have already run the AI model for these slides. But once you click on Run Model and say I click on Order Details for now, you can see that the processing has been complete. And if it was not completed, it will give you an option to sort of, look at this page. So here you can quickly look at what the AI tool is doing, like a quick mock-up demo. And with few, link to validation reports and the compatibility and things like that. And once you have done that, you can run the model directly. Now, say you want to look about this particular AI app in more details. So you can either click on view details here, and it takes you to an AI apps page that gives you access to, as much information about the app that you would like before you try out the AI model on your slides. So what you're seeing here is a very short demo of how the AIRA matrix liver analysis looks on the platform, and also what are the specific features that the AI model detects. So we have EMH, we have microgranuloma, bile duct, vacuolation, and other features which I will show in a second. It also gives you access to the vendor information, and it also gives you information about the AI app specifically, like what it was trained on, and so that you can make a perfect choice for your slides. Now, on the platform itself, we have the ability to select on applicable slides. So what this essentially means is that we will sort through the images based on the compatibility. Like say, for example, you have a liver rat across an SPS file. Now that's an applicable slide to use AIRA Matrix app. But say it belonged to a different species on which the AI is not trained on. So we don't show it over here. Again, we want to make the experience easy for you to test the most compatible AI app for your research. Now, going back to the analysis itself, I can click on any of the feature that it detects. Say I click on bile duct, EMH, microgranuloma, single cell necrosis, and vacuolation. It shows in as overlays. And say I would like to zoom in on bile duct regions. You can easily do that very rapidly at a higher resolution. And as I mentioned previously, you will also have access to quantifications. Like for example, over here, it shows that 0.2% of the entire tissue image has bile duct, spanning across over 590 regions across the image. Now, not just that, say you want to look at multiple slides simultaneously, you can easily do that on the platform. And say I click on uh, 4 slides over here. Again, it doesn't need to be 4. You can select as many slides as you would like. It can load it for you, and say you want to compare images side by side, and you also want to look at the AI result to help in your comparison. So you can do that quite easily, and you can zoom in into that specific region of interest. For example, let me go. Okay, good. So you can look at a specific region of interest and try to compare as to, what's happening across these images. Again, the experience of loading these images side by side and loading the AI result is very quick. Now, of course, to run the AI app, it will take a couple minutes. And say you are running AIRA matrix liver analysis on 100 slides, it will take some time once you click on run model, but you will be notified once the results are ready. And once it's ready, it's a matter of milliseconds for you to load those results and look for yourself. So again, you can scroll through. As you can see at the in the bottom panel, it has a lot of vacuolation, which you are seeing here on the slide, and you can also like turn off specific features that you would like, and you can interact with these AI apps. And another very interesting takeaway is that through AI orchestration, you can cross play across multiple apps simultaneously. Now, what do I mean by that? Like, say you're interested in, for example, HistoWiz tissue segmentation tool, and you're interested in AIRA matrix like liver analysis. You can, cross play across these multiple apps simultaneously. Again, this is just an example scenario. And another example would be, say you're interested in cell segmentation or, percentage biomarker quantification from QPath, and you're interested in AIRA matrix analysis. So you can load in these results simultaneously and, look for the result. And not just that, we also have the ability to upload third-party annotations directly to the platform. And again, this sort of helps to accelerate your research, because it might take some time to add new buttons, add new AI buttons onto the platform. So that's why we also have the ability to upload third-party annotations directly. Say there is another application from AIRA Matrix, and we want to know, before we create another button, we want to know how it looks on the platform, and have some customer studies that can be done. So you can upload annotations directly to the platform with a click of a button, and the results will be available. So uh, before closing in, so the high level message is, what we are trying to do with HistoWiz PathologyMap platform is create a centralized hub to manage all of your data. And again, by data, I mean not just your images, but it could be the metadata information, it could be the pathology reports, it could be AI analysis, or any other annotations, that you would like to upload onto the platform. Again, this will help you to accelerate your research, because you will have all of this information at your fingertips in one single place. And we want to create like a circular experience for you. What I mean by that is, you can literally go from anywhere starting on the platform and reach anywhere else. So you can start with uh, uploading your slides and third party annotations, or you can send us the tissue blocks and we can digitize the slides for you and give you an end-to-end experience. And with that, I am happy to take any questions.
Well, thank you. Thank you, Akash, and thank you, Nitin. Really nice presentation and a very clear demonstration of the product there. The speed of analysis is amazing and should be very beneficial in the preclinical and the toxicology market when you have so many multiple organ targets. At this point, we're going to move on to questions from the audience, and I can see that we do have a few questions that have come in today. So let's move on to the first of those. Yep. Okay, so I think this question is for Nitin. Can you calculate the transfection percentage for the tissue sample as a whole and for individual cell types?
Okay. Thanks, Chandan. We haven't specifically looked at transfected cell identification and classification. However, we have developed algorithms to detect cellular changes such as mitosis and single cell necrosis. Additionally, we have developed solutions which can identify different cell types and detect an increase or decrease of the cellular population. Say, for example, lymphocytes in spleen, or looking at bone marrow, erythroid, myeloid, and megakaryocyte. As a company, we do offer custom AI solution development, and would be very happy to discuss this further offline.
Okay, thank you. Another question here for you, Akash. Is it the technologist's responsibility to monitor quality of slides?
So yes, it's the histotechnologist's responsibility to monitor the slides, and sometimes, like monitoring the slides without using analysis tools might be difficult. And I just showed you a scenario such as like out of focus, which really requires you to go at a very high resolution to detect anomalies and issues in the slide. So AI tools help to assist in that process. So at HistoWiz, our lab uses the AI tool to sort of accelerate, producing high quality slides. But at the end, yes, I think it's both AI and a human expert who play together to produce high quality slides.
Okay. I think this one is also for you, Akash. Can we create pathology reports on the system using AI?
Yes, we can. And we didn't show you a demonstration of that in today's webinar, but we also have the additional capability to download the quantifications into a spreadsheet, into a document. We also have the ability to show graphical representation of the feature quantification. So all of that can be directly embedded into a PDF report. So to answer your question, yes, we can do that.
Okay, thank you. Another question here. Could you give an example of some real-world applications of this AI-driven liver toxicity assessment in preclinical research? Who's that one for?
Sure. So I think few of the examples which we are currently working with the pharmaceutical companies as well as CROs are the primary and the peer review workflow. So given a study done as a primary review by a CRO, these AI-driven toxicity assessment tools helps the pathologist to not just accelerate the toxicity assessment, but also to accurately identify some of the more subtle and more rare findings. When it comes to the pharmaceutical side, looking at peer review workflows, the same algorithms help the pathologist to do a second opinion read on some of these things using AI assistance, and again helps in generating more reproducible outcomes and a decision which is driven by AI and then a pathologist in the loop.
Okay, thank you. Again, I think for you, Akash, how easy is it for researchers to integrate and use AIRA Matrix's AI tools within the PathologyMap platform?
Right. I think the core focus for us through PathologyMap platform has been to provide a seamless experience with respect to the speed with which you can access these features and also load in analysis tools. So as I mentioned in today's webinar, you can see the liver analysis model as a button on the platform. And you can just click on the run model and you will be notified once the results are ready for your slides. And Nitin also mentioned about a wide suite of AI tools apart from liver analysis, which they have. And we are very eager to sort of onboard more tools based on the customer's requirement. And since we are optimizing the liver button and making sure the data and the annotations flow in seamlessly to and from the system, we will be doing that for other tools as well. So again, we are in the middle of doing a strong pilot with our customers, and the feedback has been positive. And we want to assure that we want to keep the experience quite simple for you, and we will do all the hard work of the data infrastructure, the platform, and analysis.
Okay, thank you. There's a question here. How does AI improve the reducibility and accuracy of specifically preclinical liver pathology analysis compared to traditional methods? That's probably for you, Nitin.
Yeah. So these AI algorithms is the biggest problem in, say, for example, the current manual workflow will be inter and intra observer variability. That goes in any kind of pathology assessment. How does AI overcome the observer variability is by when these algorithms are trained, these algorithms are not trained by one pathologist providing their ground truth. So it's usually a panel of pathologists, and a majority consensus is taken into account while we develop these algorithms. So these algorithms tends to be having less bias towards a single pathologist. So it is more generalized in that sense when it comes to the variability which is generated by the human read. In terms of accuracy, what these AI algorithms looks at is, since even the pathologist looks at an image at a multiple magnification, but there is a extent to a limited extent which a pathologist can look at it. So in a typical pathology tox assessment, pathologist looks at 10 high power field, but an abnormality could be there in any part of the tissue. What AI does is it actually run the analysis on the entire tissue and generate a more comprehensive assessment of the various toxicities and the various abnormalities which are present. So in that sense, the quantification which is produced by these AI algorithms is much more accurate as compared to a pathologist limited review.
Okay, thank you. Final question. What feedback has there been from early users regarding this integration, the AI integration on PathologyMap? And has there been any comments on how it's improved workflow?
Yeah, thanks for that question, Jonathan. So we have received a great amount of feedback, and I think it's mostly for the flexibility of using the tools on the platform, because our customers have been using PathologyMap to manage their slides, and I think for them it is very easy to click on like the specific tools like bile duct vacuolation and all that, and get the quantifications at fingertips. And not just that, we also have users who sort of upload their own analysis annotations, which I just showed during the demo. So I think the feedback has been in terms of like how open is the platform to ingest multiple analysis and the flexibility. And again, going forward, we just want to work more on how do we automate the report generation. I think this was one of the question by the audience on generating pathology report. Right now, the report is like, you can access the quantifications and graphing, but I think we can convert that into, natural language to have a complete report based on the analysis and based on your annotation. So we want to integrate tools on the platform which can ingest not just the analysis result, but the metadata information, the comments or annotations, custom annotations on the platform, and have it as a report. So, but again, you will be seeing much more improvements in the coming future.
Great, well, thank you very much. And that brings us to the end of the questions. So just time for me to thank you both. Thank you, Akash. Thank you, Nitin. A really lovely demonstration and a really interesting presentation from both of you. So thanks for your time today. And to our audience, thank you for being with us. I hope that was entertaining for you. Just to tell you again, obviously, you can look at you can follow Pathology News looking at both companies, at their pages. We've got lots more information. And thanks for being with us, and we'll see you on the next webinar. Thank you.
Thank you, everyone.
Thank you.